right, so our final speaker of this session is Sergey Velyichko from Soxigen. He's going to tell us about generating stem cells with greater developmental potency using Super-SOX.
I have the slides. Thanks for the introduction. So I did my PhD at Max Planck Institute with Hans Scholler, who has discovered OCT4. And I did the postdoc with George Church at Harvard Medical School. And just a few months back, I started the company with the goal to generate stem cells capable of building whole bodies, whole human bodies. So the dimer that you can see on the slide, this is OCT4 and SOX2. These are the two transcription factors, two Yamanaka factors that are responsible basically for resetting life in every generation of vertebrates from fish to human. This is what basically makes babies born young. So Shinya Yamanaka made this huge discovery, discovered iPS cell technology, induced pluripotent stem cell technology, and that enabled derivation, very cheap and easy derivation of autologous embryonic-like stem cells for people, for patients. But for fundamental science, I think the very interesting breakthrough, the important breakthrough is that Yamanaka reduced the question of what is developmental reset to just understanding these four factors: OCT4, SOX2, KLF4, and c-MYC. This is the only technology that we have that can rejuvenate all cells all the way back to the youngest cells that ever exist in the body. So pluripotent stem cells can exist in a few different states. It's like a range of states, and most prominently it's naive versus primed pluripotency. So naive pluripotent stem cells represent pre-implantation embryo, so before the embryo implants into the uterus. And the naive stem cells can still build whole bodies, while the primed cells represent post-implantation embryos, so later partially differentiated state, and they lose this ability to self-organize into whole bodies. So for the mouse, we have both states isolated, and we can grow stem cells in both states, but only naive stem cells can make mice, while primed cells cannot. But for all other species, even if you take pre-implantation embryos and culture them, the stem cells are stabilized at later stage. So basically, they cannot build animals. So the most convincing proof that the pluripotent stem cells are truly pluripotent is creation of whole bodies. And this can be achieved using tetraploid complementation. And tetraploid complementation is the method that starts with a two-cell embryo, which is electrofused to create a tetraploid embryo. And this tetraploid embryo can generate functional placenta, because mammalian placenta can tolerate being tetraploid, but it fails to build the body. So basically, these tetraploid embryos have an empty niche for the whole body, which can be now complemented by iPS cells or ES cells to be tested. And the true pluripotent stem cell should be able to create complete animals. And this has been achieved for mice, and that's it, basically. It doesn't work for other species. No livestock has been generated, no primate has been generated entirely from stem cells. And the second best proof for pluripotency is the so-called germline transmission. This is the ability of stem cells to contribute to the body, not necessarily build the whole body, but at least contribute in development to the body, and particularly creating sperm and egg, functional sperm and eggs that can build the next generation. And this has been achieved for iPS cells. It has been achieved for mice, for both males and females, and only in one paper, only once it has been achieved for male rat iPSC. So this is the current state of iPS cell field. It's very limited to mice, and even for the mice, it doesn't really work so well, and it doesn't work for all the breeds of mice, only some breeds of mice. So 2 years after his Nobel Prize-winning work, Yamanaka published this Nature Biotech paper where he dissected his cocktail. He removed each of the component one by one or replaced it with other members of the family, of the same transcription factor family. And what he found is that MYC is the least important of his cocktail, so you could completely remove it and still generate iPSCs. SOX2 and KLF4 can both be replaced by the other members of the same family. So SOX2 can be replaced with SOX1 or SOX3. KLF4 can be replaced with KLF2 or KLF5. But OCT4 is the only Yamanaka factor that could not be replaced. And therefore, the conclusion was that OCT4 is the most irreplaceable of the Yamanaka factors. So I published this paper on the cover of Cell Stem Cell that showed that it was actually wrong, that we can remove OCT4 and we can generate SKM iPS cells in mice. But the most exciting data was that if you omit the OCT4, the factor that was thought to be the most irreplaceable and the most important Yamanaka factor, you get this iPS cells that are 20 times better at making mice comparing to the original OSKM iPS cells. So we have achieved the highest efficiency of stem cell derivation of complete animals with more than 80% of live births. This is also the highest efficiency of mammalian cloning for any species ever reported. Yeah. So these are the data. You can see that the original OSKM iPS cells are pretty bad at making mice. Some pups are born, but they usually die within a few days after birth. While if you remove OCT4, you can get SKM iPS cells that all the lines could make pups. So each dot here each data point represents the average survival of embryos created from a given line. And so this is a lot of data condensed in one graph. You can see that most of the lines, when you remove OCT4, can generate adults, and that's nice. So this was recognized as a top technical advance of 2019, and Yamanaka himself was interviewed, and he commented that if we could achieve this for human, it would be a huge advantage for iPS cell based therapies. And indeed, 4 years later, we have achieved this for human using what we call Super-SOX. And Super-SOX is a point mutant, is a chimeric factor that is partly SOX2, partly SOX17. So both SOX2 and SOX17 in early development cooperate with OCT4. When SOX2 kind of wins over OCT4, they bind together on the canonical SOX-OCT motif, and that induces the future body, the epiblast that makes the body. If SOX17 wins, the OCT4 is recruited to so-called compress SOX-OCT motif, and that induces the yolk sac fate. So we kind of combine the best parts from SOX17 and SOX2 to create this enhanced SOX2 molecule that has much higher stability with OCT4 on DNA. That globally redistributes SOX and OCT binding to the canonical SOX-OCT DNA that controls all of the pluripotency genes. And that enables us to create better quality stem cells even in the presence of OCT4. So how did we come up with this? This was actually a basic science project. During my PhD, I was trying to answer the question that was posed by Yamanaka in his Nature Biotech paper. And the question was why OCT4 is unique among the OCT factors, why only OCT4 can generate iPS cells but not other OCT factors. And what I kind of accidentally found is that if you swap certain parts from SOX17 into SOX2, particularly this one residue at position 61, if you change it from alanine, which naturally exists in SOX2, to valin, which naturally exists in SOX17, you can enable generation of iPS cells with other OCT factors, including the ones that Yamanaka himself showed to be incapable of making iPS cells, like OCT2 and OCT6. So this was very interesting. This means that, like, without touching OCT4, you can OCT factor, you can enable reprogramming, which means that the true unique property of OCT4 must be that its ability to cooperate with SOX2. So the most exciting data from that study was, again, the tetraploid complementation experiment, so creation of all-iPS cell mice. And you can see that this single amino acid swap that we know exactly what it's doing, it enhances the dimerization with OCT4, can enable generation of adults. So Super-SOX, so this was point mutant. Now this is not just the point mutant, but also we introduce other pieces from SOX17 to make it even more potent. This is what we call SOX217. It's basically half SOX2, half SOX17. It enables generation of iPS cells much faster, even in just 1 day of induction. So these are the fastest iPS cells ever made in just 24 hours of induction, and they already can make mice and contribute to the germline. It also enabled us generation of iPSCs for new species that are commercially valuable, like cow, pig, cynomolgus, macaque. These are the first iPS cells that are integration-free, generated using episomal vectors. And it also enabled generation of iPSCs for particularly older patients, where sometimes it's very inefficient. But the most exciting finding is that even regardless of Super-SOX, so in this slide no Super-SOX was used, the stem cells that are capable of building bodies naturally have higher levels of SOX-OCT dimer. So we could lyse the cells and measure the content of the dimer by just running so-called EMSA experiment, the electromobility shift assay, where you just mix the lysate with target DNA and run it on the gel. And you can see that the naive lines that we know can make mice, marked with green, they both have, so they have higher content of SOX-OCT dimer, and the 2I media that actually was discovered around 15 years back, and that was the media that enabled generation of mice that were called so-called non-permissive mice. So before it was just one breed of mouse that we could make, and the 2I media enabled making other mice, and for the first time enabled another species, which is rat. Nobody knew how exactly it worked, especially one of the components of these two inhibitors. But we showed that it enhances the OCT-SOX dimer. You could also see it in the published data from somebody else, and this is ATAC-seq data of naive cells being exposed to FGF, which makes them primed, so basically partially differentiate them, makes mouse cells more like human cells, and you can see that the most lost open chromatin marks are SOX-OCT dimer of all the transcription factors. We also found a cocktail that could enhance this dimerization and could convert regular human iPS cells into naive, so representing pre-implantation embryo. And this is episomal expressions of Super-SOX and KLF4, so the subset of Yamanaka cocktail without OCT4 and MYC. So KLF17 and SASD2 are the genes that are only expressed in human pre-implantation embryo, but not post-implantation. You can see you can activate them using these plasmids, and interestingly, somehow this conversion also eliminates the plasmid itself. It's marked with M cherry, and you can see it disappears. You can see that also on sorting data, where the control could not generate any SUSD2 positive cells. SUSD2 is a surface marker which can be used to sort the cells. And while the MCherry SK could generate MCherry negative SUSD2 positive cell population. So this naive cells expressed naive markers, and again, eliminated the episomes. We also achieved this using mRNA and using feeder-free culture and using regular commercial human stem cell media. And the nice feature of this reset is that could be useful not just for people who are interested in human naive cells, but for anyone interested in pluripotent stem cells, is that it enables passaging cells as single cells without ROCK inhibitor. So ROCK inhibitor is apoptosis inhibitor, and one of the difference between mouse and human cells is that mouse cells are very clonogenic. They can be easily passaged as single cells and survive. Each of them will give a colony, while human cells will all die if you passage them as single cells unless you block the apoptosis. So the question is, can these cells make babies? Of course, I could not answer that. It would be illegal, but what we've done here is we aggregated these human naive cells with mouse embryos, and we could see that the cells that are marked with RFP, human iPS cells that are marked with RFP, were integrated in mouse inner cell masses, so the parts that will make the body. And in some cases, they could overtake the whole epiblast part. The epiblast part is what becomes the future body, which is pretty cool. So this is the embryo that has the epiblast part from human, but the future yolk sac and the future placenta, the trophectoderm, is from the mouse. It also works on other species, so we tried cow iPS cells. So the cow iPS cells are so in the range from of pluripotency, they're even lower than human. They're so-called loser cells, as opposed to winner cells that are like mouse cells that are clonogenic. They survive, they engraft. So the cow cells cannot even cow iPS cells cannot even make teratomas if they're injected in immunocompromised mice. But after the treatment with the episomal vector that we just showed the data for human, they could engraft and create teratoma. We also showed, in collaboration with a Church Lab PhD student, MingCeler, we showed that this treatment of Super-SOX and KELA4 can enhance germline production for humans. So this is differentiation of human iPS cells, 3 different human iPS cell lines, including one derived from George himself, into oogonia-like cells to create precursors of eggs. And yeah, you could boost the efficiency of that, and as well as produce cells that are more demethylated, more similar to the original oogonia cells derived from ovaries. So the committee asked me to present in vivo reprogramming data. We don't have any yet, but we have generated these mice that have actually using Tetraploid complementation, so much faster than normal. Could generate immediate homozygous edited animals from stem cells within just 2-3 months. And this pups are carrying that-inducible Super-SOX cocktail to attempt whole body rejuvenation. Very risky, but would be interesting to see what happens to them, to the mice. So this is the take-home message. So we could reset regular human iPS cells using mRNA into more potent human iPS cells, more clonogenic, more capable of creating tissues, and potentially with enhanced engraftment and functionality. So these are the people who contributed to the project. Most of it was done in Max Planck Institute and funded by ERC. And also, I want to thank my current funders, venture capitalists. Thank you.
Thanks very much. We have time for a few questions. Maybe I could ask the first one if you don't mind.
Yeah.
Do you have any idea about what the transcriptional programs are that are induced with overexpression of OCT4 that prevent pluripotency?
We have studied that, and yeah, we have. So we have quite some data. It appears like when you overexpress OCT4 in fibroblasts, which are normally not expressing OCT4, the binding profile is very different from what it is in embryonic stem cells when it naturally expressed. So naturally, it binds around 6,000 sites in the genome, but when you express in fibroblasts, it binds like 200,000 sites. So it's all over the place, and it kind of starts imitating other OCT factors that are tissue-specific. So binding to particularly skin-related sites and generates kind of like epithelial-like cells rather than pluripotent cells. But also naturally, pluripotent state is not supposed to last, right? Naturally, it's very transient, and what you want is, you don't want to become, to be the embryo forever, right? You want to become, you want the cells to differentiate. And OCT4, as well as SOX2, both have double functions. So as a monomer, it is responsible for priming and differentiation. As a dimer, it resets the cell fate. Similar with SOX2, SOX2 alone binding is inducing neuroectoderm differentiation. So all of these factors evolved to have multiple functions, and what we kind of enhancing here is this specific one function, which is reset.
Thanks. A question over here.
Thank you. It was very interesting work. I wonder, cancer stem cells or cells with tumor initiation capacity, they overexpress exactly all Yamanaka factors. So I wonder if your Super-SOX animals later in life will start developing tumors earlier or faster. Did you have a chance to observe this and monitor these animals long enough to see what later in life they will have more tumors than regular animals?
We have some animals that lived long life, and we never saw any tumors. So we were not overexpressing those factors, right? We created those animals from that inducible or episomally derived iPS cells, and then we turn off the transgene, right? So we have not yet tried to overexpress them in adult bodies. That might indeed lead to tumorigenesis. We will see. But I don't think any cancer overexpress all the Yamanaka factors. There are certain cancers that overexpress some of the Yamanaka factors, but I don't know if
CIMIC is a common
CIMIC,
driver.
yes, but we are omitting, yeah.
But also many cancer stem cells overexpress SOX2.
SOX2 is true, yeah.
And
Glioblastoma is one that is famously expressing SOX2, and the more SOX2 you have, the more likely you will die from glioblastoma. I don't know though if any of them overexpress all three of them, excluding MYC. MYC is very oncogenic.